Appendix is a common topic. Does appendix has an artery? Appendicular artery is very much there. And it is a posterolateral wall of the cecum about 2 centimeters below the ileocecal orifice. It has a mesoappendix very much. If you become a laparoscopic surgeon, you have to somehow go through the keyhole and separate that mesoappendix is a very important challenge. Appendicular artery is a branch of the ileocolic artery is very important to remember. They are very much tinea coli, absence of them is false. And typically those tinea coli will proximally converging at the base of the appendix, and from where the cecum and appendix arise, doctor. It is the anti-mesenteric border of the midgut loop, not mesenteric. That is a point from where they arise. One question on brachial plexus is routine. Axillary now this time. What does it innervate? Deltoid and teres minor, whereas biceps brachiae, coracobrachialis are by the musculocutaneous, and teres major part of which is not a part of your rotator cuff. What does rotator cuff has got? Sits. T is minor, not major. Okay. So subscapular is the one which is innervating the teres major. Where do you check for the axillary nerves? Cutaneous innervation, lower half of the deltoid. So do you want to, if you are suspecting a fracture of humerus, axillary nerve damage, do you want to check motor function or cutaneous function? Cutaneous, because abduction to test in a fracture patient is criminal. So there is a reason cutaneous, lower half of the deltoid is very important for axillary. Now let us talk about the parotid gland. Typically the duct is 5 centimeters long. Along the anterior border of the gland it will be emerging. Upper second molar tooth is the point where it opens into and it pierces the buccopharyngeal fascia, buccinator muscle, and the buccal pad of fat is what need to be remembered. Now a very important question. Auriculotemporal nerve is the secretomotor fibers which will go to the parotid gland. That's the reason if you do parotidectomy there can be aberrant innervation of this auriculotemporal onto the overlying skin, which is called Frey syndrome, as all of you know very well. But auriculotemporal is a branch of which nerve? Typically it is facial or is it the mandibular? Basically mandibular of the trigeminal nerve has got 5 important, certain important branches. You have the auriculotemporal branch, here you have the lingual, inferior alveolar and buccal, mental and nerve to mylohyoid. So you must be very sure 5th to 7th cranial nerve, doctor, they are like the gods for the entrance exam. Now from where does the salivary arise? Basically it is a junction between the ectoderm of the stomodium and the endoderm of the foregut where it is arising from. That's the reason it has a origin from both ecto and also endoderm. And let's go to the next question. Anterior abdominal wall, rectus sheath, how is it formed of, and external internal oblique muscles. This we discussed in Ames Jan paper also. The linea alba is the raphe, which is the central part, which is avascular. It extends from the xiphoid up to the pubic symphysis, but most important to observe is below the level of the umbilicus it is very narrow and in fact very difficult to define. It is only thickest above the level of the umbilicus. That's what we need to realize about the linea alba, which is poorly formed typically in the lower 1/4 of the anterior abdominal wall, is what I like to underscore. Now rectus sheath, the composition of it is different at different parts of our abdomen. Typically it is the arcuate lines which will define that. Above the costal margin, below the costal margin and up to the arcuate and below the arcuate. That's how we look at the rectus sheath. Typically the posterior layer of the rectus sheath is incomplete and deficient below the level of the arcuate line is what we have to clearly remember. Now if you take the rectus muscle, it has got 3 transverse fibrous bands. The reason is it is not built as a single muscle or developed as a single one. It is 3 pieces which is joined together. So it is typically a segmental origin of the muscle which is classically reflected in that. Now let us talk about the external oblique muscle. Is it same throughout? No. In the lower 1/4 of the anterior abdominal wall it becomes aponeurotic and inguinal canal anatomy. It contributes, all of you know very well. Third cranial nerve, supplies everything but not superior oblique, and lateral rectus by abducens and superior oblique by the trochlear, as all of you know. But tell me, doctor, 4 important unique features of trochlear, favorite question of examiner. It is the smallest nerve and it is the greatest length, intracranially it will be passing through. It is the only cranial nerve which will be decussating even before innervating its target. Nuclear level only it will decussate and come out. Others will reach just about to, for example facial, just about to innervate the muscle, they undergo the decussation. But at the level, nuclear level only it will decussate. And it is the only nerve which comes from the dorsal aspect of the brainstem, as all of you know very well. Isthmus of the thyroid, which is important if you are doing any tracheostomy, it is very important. So that's the reason it is between the 2nd to 4th tracheal ring, typically where the isthmus of the thyroid is located, is what need to be remembered. Now superficial perineal pouch, and what are the muscles of the superficial perineal pouch is a very important question. Basically we talk about the superficial perineal pouch. What are the boundaries of it? The inferior border of the perineal fascia and the superior border of the perineal membrane, and it has got the 3 important muscles: ischiocavernosus, bulbocavernosus, and superficial transverse perineae. These are the 3 muscles located in that. And the crura of the penis, bulb of the penis, and the greater vestibular glands in the case of the female, they are also the components. So that's the reason, doctor, out of all, it is the bulbospongiosis which is the component of the superficial perineal pouch is what need to be remembered. In our anatomy regular discussion, we discussed on deep and superficial perineal pouch in a greater detail. Now intercellular receptors, what are they? What are the secondary messengers associated? And what is the structure of insulin receptor, which is a favorite topic of examiner? Let us talk about that. The nuclear receptors located in the cell nucleus, and in the endoplasmic reticulum the IP3 inositol triphosphate receptors, they are all intracellular. They use the inositol triphosphate, and lot of hormones use these intracellular receptors. For example, steroid hormone receptor, thyroid hormone, retinoic acid, and liver X receptor, etc. They are all transcription factors which are intracellular receptors. Similarly, IP3 receptor is intracellular. Now let us talk about insulin. Insulin is not intra, it is transmembrane receptor. There is some part intra, some part extra, and it has got a tyrosine kinase activity by itself. And the glutamine, GLUT4 is the receptor where the glucose will be causing the one which is making an entry by the action of the insulin. Now let us talk about insulin production. Every time insulin is produced, one C peptide is produced. So you can know whether it is a factitious insulin administration responsible for hypoglycemic episode or an insulinoma responsible. How do you know? High C peptide along with hypoglycemic episodes is indicative of an underlying cause, endogenous insulinoma to be responsible. Now let us talk about the ion composition. All of you know potassium is typically intracellular and you have a lot of anions, whereas the chloride and sodium are more often extracellular is what need to be remembered. ADH, antidiuretic hormone, is a favorite topic of examiner. We need to talk about it. If there is no ADH, the collecting duct is not permeable to the water at all, and large volume of urine become produced. If there is ADH, then it becomes permeable and only small volume of urine is produced, all of you know very well. Then who produces ADH? It is typically from the posterior pituitary it becomes released, so it is produced in hypothalamus, and it is the high blood osmolality which is the stimulant for the production of the ADH. Now doctor, when do you require ADH? When you have to retain water, that means you don't have water. Whenever osmolality is very high, you are dehydrated, you require ADH. ADH production has 2 stimulants. The hypovolemic state could be one reason, second is osmolarity. Common questions asked in the exam are, what level of osmolarity is required, and is it osmolarity more important, or is it the hypovolemia, I mean volumic status, which is more important? Let us talk about it. The osmoreceptors are located in the anterolateral hypothalamus, which we have seen in earlier graphic, and it is below the level of 280 milliosmoles per kg of osmolality. Virtually ADH is not present at all in our plasma. The moment even a small change is there, 1% change also, more than 290 if we cross the milliosmoles of osmolarity in the plasma. What is normal plasma osmolarity? 280 to 290, let us say. More than 290 is a strong stimulant. Then similarly, if you take the urine osmolarity, typically a urine osmolarity of 1250 milliosmoles per kg results in a similar kind of antidiuresis to happen, is what need to be basically remembered. Now a very important question comes here. A osmotically active substance must make ADH to become secreted. What are the two common osmotically active substances that we come across? Sodium, urea also. Is urea also capable of stimulating hypothalamus to produce ADH? No, because urea cannot cross the blood-brain barrier and reach hypothalamus, only sodium can do. Hence one of the common questions. Which osmotic substances are capable of crossing blood-brain barrier and stimulating the anterior hypothalamus to produce? Now the second stimulation and control of ADH is by the baroreceptors. Where are they located, doctor? Typically carotid sinus, aortic arch, they constantly will be knowing what are the pressure changes, and the sensory fibers from them will pass to the cranial nerve 9 and cranial nerve 10, and baroreceptor is one strong stimulant. But the question is, baroreceptor is very lazy. That is just 5% change of the hypovolemia occur, immediately ADH won't secrete. Whereas in case of osmolarity, 1% change also will make ADH to become secreted. More than 15% of the fall of the volume and fall of pressure is required to stimulate the ADH, is what need to be basically be remembered. But the rule is, if at all the baroreceptor is stimulated, the amount of ADH released is more than what osmolarity stimulates. So for a chronic management of ADH stimulation, it is osmo important. For an acute scenario where high volume of ADH is need to be pumped out, you require baro, which is stronger. Any point of time baro is stronger, it can override even the osmo stimulation. Now similarly, drugs like nicotine, morphine, barbiturates, they increase ADH, whereas alcohol inhibits. That is the reason at the end of a binge party, when we take a lot of alcohol, no ADH, so we go into diuresis and we need to go into the toilet. So anterolateral hypothalamus is location for osmo, carotid and aortic sinus for baro. Plasma osmolarity for osmo is 1, circulating volume for baro, and 10 to 15% fall is required for the baro, only 1% above 280 is sufficient for osmo, and amount of ADH released because the baro stimulation is more than that of osmo, and in fact baro can override that of the osmo stimulation, is what need to be remembered. Fine, ADH got released, then what will it do? It acts on the collecting duct on the receptors, it also is capable of acting on the vessel and cause vasoconstriction. Common trickling question in exam is it V1 here or V2 there? So we must be very sure, precision is entrance, right? So it is the V2 receptors on the basolateral membrane where the activity coupled with the adenylate cyclase and G protein cyclic AMP dependence is the mechanism by which water is being reabsorbed by the action of the ADH. So V2 receptors are important for the collecting tubule action of the ADH, and which is cyclic AMP mediated, is what has to be remembered. Whereas you have the vasoconstriction, which is the V1 receptors on the vascular smooth muscles, is what I like to underscore to all of you. Now let us go to the next question. Kidney produces what important hormones? All of you know erythropoietin and also vitamin D is active form, and renin, they are all the ones which are being produced. Now what is axonotmesis? If you go to the deja vu of AIMS November 2007, recently we are talking about this topic. We must be very sure. After all, nerve is what? Nerve is one copper wire and one plastic sheath. You have an axon and you have a myelin sheath around it. Damage to both, only to myelin sheath, only to axon. There are the different possibilities. Accordingly, Seddon, the great man, has classified it into 3 types. So axonotmesis is axonal damage and also the myelin sheath damage, but there is a preservation of endoneurium, perineurium and epineurium. So motor, sensory, autonomic, all of them basically will undergo the crash, and axon can in fact regenerate, and that can lead to recovery. So we need to remember we have neuropraxia, axonotmesis, neurotmesis. In neurotmesis, both the sheath and axon are gone. Sheath is relatively still preserved well. In axonotmesis, the sheath is well, but axon is disrupted. Whereas in neuropraxia, neither of them has lost its continuity. So neuropraxia stands the best prognosis, as all of you know very well. Now collagen, different types, where do you find? It is like multiplication table in childhood. Till what table learning in multiplication is correct, doctor? 9 is sufficient? Or do you need to read up to 25? 25th table. I still find, we used to be bad in maths, but my friends who read the 25th table better finally have become something different. I mean, our knowing multiplication table in high school really does not decide how successful we are going to become. That's what we learnt at a later part of the day. But it used to be a tough time to convince Granima that I am also still intelligent, though I know very less number of tables than that fellow. So basement membrane, collagen numbers are also same, doctor. You have 1, 2, 3, 4, 5, 6, 7, 8, 9, 12 until 23. So which place are they each located? Difficult to remember. At least up to 9th table we will learn. Okay. So number 1 is present in scar tissue, endomysium of the myofibrils, tendons, Ehlers-Danlos because of the type 1. Type 2 is in hyaline cartilage, and type 3 is in Ehlers-Danlos once more. Granulation tissue, it is important, and also reticular fiber. And in basement membrane of the kidney, what you have is basically type 4 in the glomeruli of the nephron, and Alport syndrome is because the type 4, that's what examiner asked. In kidney, what do you find? Type 4. Then type 5 is seen in placenta, and there is one myopathy on the name of type 6. Type 7 is in epidermolysis bullosa, in the dermoepidermal junction it is typically found. Type 8 is in endothelial cells, and type 17 is once more interesting because it is there in bullous pemphigoid implicated. So at least type 4 is in GBM, glomerular basement membrane, that much is more than enough. Now what are the functions of the limbic system? Let us talk about it. It has got hypothalamus, hippocampus, amygdala, limbic cortex, 4 components of the limbic system. Out of this, the hypothalamus, reward and punishment, vegetative functions, temperature, hunger, thirst, libido, etc., etc. Behavioral control, sexual drive, etc. Endocrinal function, etc., etc. Now coming to hippocampus, the rage, pleasure, excessive sex drive, passivity, learning also, unfortunately. So all of them are in the same place, that's the problem. Hippocampus is the part of the limbic system. Then you have amygdala, olfactory stimuli and interrelationship from one part of brain to the other, you require amygdala. And next you have limbic cortex. In the limbic cortex, doctor, this is very important. You have the anterior part of the temporal lobe. The cortex of it is a part of the limbic cortex. And you have a small gyrus called parahippocampal gyrus, which is also a part of the limbic cortex. Then you have got a cingulate cortex, part of the limbic cortex. Now they have different functions. It is the gustatory olfactory association with the anterior temporal cortex, and parahippocampal gyrus is the one which is associated, which will connect the temporal lobe with the other part. with the hippocampus. The connecting bridge is the hippocampal gyrus, and all our auditory association areas and etc., they are all important. Is this parahippocampal gyri is what need to be remembered. Now, what are the markers of plasma membrane, endoplasmic reticulum, lysosomal membrane we must be sure about? Nucleotidase, sodium potassium ATPase, adenyl cyclase for plasma membrane. G6 phosphatase is for endoplasmic reticulum. Then all of you know Golgi apparatus is the packing zone. That's the reason mannozidase, sialyl transferase, galactosyl transferase, everything transfer packaging is all Golgi. And ATPase synthase is an inner mitochondrial, glucokinase is basically a cytosolic enzyme is what need to be remembered. Now, hemoprostatic group, all of you know myoglobin, hemoglobin, cytochrome oxidase, all of them basically contain it. Even catalase, even tryptophan pyrrolase, they contain the hemoproteins is what need to be remembered. Now, from which amino acid do you have the origins of the creatinine is a very important question. Basically, L-arginine becomes the guanidoacetate, and during that process ornithine will be generated from the glycine, for which you require one aminotransferase. Guanidoacetate in turn become converted to creatine by the action of the transmethylase, where methyl group is transferred to guanidoacetate. Creatine typically can become phosphate, or it can undergo dehydration, can lead to creatinine and finally excreted. So, you require glycine, you require arginine for the formation of the creatine is what need to be remembered. Citric acid cycle you must be obsessive to remember, which is substrate level phosphorylation, which is NADP produced, FADH2 produced. These are all topics hanging around our neck from +2 level, doctor. So, you must be sure about it. Let us take up this game. First time when NADH become generated is in the isocitrate dehydrogenase step, where the isocitrate becomes the oxalosuccinate, and oxalosuccinate become alpha ketoglutarate. Finally, isocitrate becoming alpha ketoglutarate. It generates the NADH. Then you have the alpha ketoglutarate dehydrogenase, another fellow, who will also be producing NADH. But there is one step where FADH2 is formed instead. That is a common question in the exam. So, we need to remember it is the succinate dehydrogenase where FADH, only 2 ATPs are the final outcome of it. Then which is the only step where substrate level phosphorylation, no waiting until electron transport, directly ATP is generated. It is at the level of the succinyl CoA synthase. This is the step where directly substrate level it is produced. Then you have the malate dehydrogenase, which also will generate the NADH. So, quickly summarize, doctor, isocitrate dehydrogenase and malate dehydrogenase are the two steps where NADH is generated is what need to be remembered, and substrate level phosphorylation is at the level of the succinate dehydrogenase. Now, let us conclude the today's session, doctor. 1 carbon transfer reactions for them, which is the coenzyme and vitamins which are required. 2 vitamins are required. One is folate, other is vitamin B12. Very easy to stop the discussion at that stage, because if you go too much into it, there are a lot of things to know, but still we will go into it, because you have to apologize, sometimes obsession for details. 1 carbon transfer reactions, you require folic acid and vitamin B12. Because these vitamins come in multiple places, doctor. In hematology we talk about it. Unexpectedly some drug will cause folate deficiency, why it is causing. So, there is a reason we must know everything about them. Basically, if you talk about folic acid, it is a composite molecule. It has got one pteridine, one glutamate acid, and para-aminobenzoic acid. The combination of the 3 is basically the folic acid. Now, the thing is, folic acid, in order to participate the 1 transfer reactions, first of all by itself it need to undergo certain modifications. Who will do those modifications to folate? It is the NADH who will do that. NADPH will reduce the folic acid 2 times and it will convert the Miss folic acid into Mrs tetrahydrofolate and the contribution goes to NADH. Once it became tetrahydrofolate, now it is ready to receive 1 carbon, donate 1 carbon. So what is 1 carbon? Methyl is 1 carbon, acetyl is 2 carbons, right? So this tetrahydrofolate must receive this 1 carbon from somebody. So who is that somebody? Usually serine is the fellow who says, please come tetrahydrofolate, I will give you 1 carbon. There is another fellow called glycine who also will be a contributor for that methyl group to this tetrahydrofolate. So from both of these amino acids it will receive the 1 carbon. Now after receiving the 1 carbon, this tetrahydrofolate fellow can exist in 3 different forms. What are the 3 forms in which it can exist? It can be N5N10 methylene tetrahydrofolate by receiving it, or it can be oxidized by the NADP so that it becomes methinyl form, or it can become methyl form. So what are the 3 forms of the tetrahydrofolate which is already received 1 carbon, pregnant with it and about to deliver it? It can be in the form of methylene form, methyl form, methinyl form, in any of these forms. Fine. Now tetrahydrofolate is having 1 carbon, sir. Who want it? Basically nucleotides for their biosynthesis require 1 carbon. They will be waiting for it. So it has to deliver. So methylene form is the form in which 1 carbon is being donated at the time of the thymidine nucleotide biosynthesis. That's the reason we require folates for the nucleotide biosynthesis is what need to be remembered. So that is all the story about folic acid. You are very sure it is the 1 carbon donor. Let us talk about the vitamin B12. Vitamin B12 is very important because it is a coenzyme for an enzyme called methylmalonyl CoA mutase. Now why do we require this great long named enzyme? The reason is, if the valine or isoleucine, methionine or threonine or odd chain fatty acids, any of them, if they undergo a metabolism, doctor, they form a peculiar end product called propionyl CoA. It is an undigestible fellow. To digest that, to incorporate that and to use it, we require basically 2 important vitamins. One is biotin, other is vitamin B12. So this propionyl CoA fellow is an end product of who? Not just the fatty acid bit oxidation, even for the amino acid bit catabolism also this fellow become generated. He is an abnormal fellow, psychotic. So valine, isoleucine, methionine, threonine will produce propionyl CoA and even odd chain fatty acids produce. Then this propionyl CoA should be converted into methylmalonyl CoA by the action of the enzyme which forms that, require biotin. Then after methylmalonyl CoA is formed, it need to be donated 1 carbon unit and then this methylmalonyl CoA become satisfied and becomes succinyl CoA. He enters into tricyclic acid, that is citric acid cycle. If biotin is not there or vitamin B12 is not there, then that leads to the accumulation of the methylmalonyl aciduria can be there in vitamin B12 deficiency, or if any of them is not there, propionyl CoA start accumulating in the body. Now what is the role of this vitamin B12, for which enzyme, other than this, where else is it required? One is for converting methylmalonyl CoA into succinyl CoA. Second place, where is it required? It is also required for the methionine biosynthesis. It is important. Why? Because typically there is one cobalt ion in the cyanocobalamin, that is the vitamin B12. This cobalt ion in the 6th position will be receiving the methyl group. It will hold it and then donate whenever it is required. So that is the unique selling proposition, a unique point about vitamin B12. So the vitamin B12 becomes methyl vitamin B12 by accepting the methyl group from where? From the methyl tetrahydrofolate, which become tetrahydrofolate and donate it to vitamin B12. In turn it redonate that methyl group and help the homocysteine to convert into methionine. So that is the reason methionine synthase requires the vitamin B12's help in order to acquire the methyl group is what need to be remembered. Not only for this, even for the modification of the cellular RNAs, for the biosynthesis of the lipids, where a lot of methyl transferase reactions occur, we require extensively the presence of the vitamin B12 and the methyl group which is being donated by it. Now let us quickly examine, doctor, after the methyl group is donated by the vitamin B12 from the homocysteine to the homocysteine and that became methionine, then how will the methionine will further carry that methyl group? Basically methionine by itself cannot donate that methyl group which it acquired from vitamin B12. It has to become something else. It has to become S-adenosyl methionine. So there is a reason, doctor, methionine acquires the adenine group and become S-adenosyl methionine. S-adenosyl methionine donates its methyl group wherever it is required and become S-adenosyl homocysteine, and in turn the homocysteine once more receives from the cyanocobalamin and regenerates the methionine. That's how continuously the tetrahydrofolate, methylene tetrahydrofolate gives methyl to B12, B12 gives methyl to homocysteine, that becomes the methionine, methionine basically becomes S-adenosyl methionine, and whenever it is required it further donates the methyl radical whenever it is required. So that brings the completeness to the answer. Folic acid and vitamin B12, they are important for the one carbon transfer. Stop codon, start codon, initiation codon is one of the important topics for the exam. UAA, UAG and UGA are basically called as the stop codons, and AUG is called the initiation codon, as all of you know very well. Now the question is, this initiation codon, that is the start codon, AUG, basically codes for which amino acid? It is for the methionine. That is the reason a very large number of proteins, if you take their structure, they have the methionine as a constituent as the first amino acid, because the AUG fundamentally codes for the methionine is what you need to basically remember. Now the three stop codons, UAG is amber, UGA is opal, UAA is ochre, is the three important stop codons need to be remembered. Now in which genetic disorders it is the trinucleotide expansion is the underlying pathophysiology at the genetic level. Fundamentally, why a set of 3 nucleotides keep on repeating and that excessive repetition which is unwanted, why does it lead to pathology is a very important issue now. There are some important unstable areas called the microsatellite repeats. It is the excessive replication and reading of them is the one which is responsible for this abnormal proliferation is what we need to basically understand. Now there is a very important phenomena called genetic anticipation is what we see in the case of trinucleotide repeat associated disorders. What does it mean? As the generations pass, the number of trinucleotide abnormal repeats keep increasing, so the severity increases as the generation passes, which is called genetic anticipation is the phenomena which is typically seen in the case of the trinucleotide repeat associated disorders. Now there are 2 important groups of the trinucleotide repeat associated disorders. One are called the polyglutamine disorders, other are called the non-polyglutamine disorders. Huntington's disease, spinomuscular atrophy or Kennedy's disease, spinocerebellar ataxia of the type 1, 2, 3, 6, 7 and 17. These are the variants which are typically called the polyglutamine type of trinucleotide repeat associated disorders. Whereas the Fragile X syndrome, Friedreich's ataxia, myotonic dystrophy, spinocerebellar ataxia of the type 8 and 12, Typically they are called the non-polyglutamine type of trinucleotide repeat disorders is what we need to remember. Then how about the amyotrophic lateral sclerosis is my question to all of you. Fundamentally, what is the genetic defect in amyotrophic lateral sclerosis, otherwise called motor neuron disease, doctor? Which enzyme it is? Superoxide dismutase is the one which is abnormal. Normally superoxide dismutase will detoxify the oxygen free radicals. If it is abnormal, oxygen free radicals become abundant and they lead to development of the neurotoxicity, which you see in the case of the motor neuron disease, is what need to be basically remembered. Now let us take up the next question. Which is the area which is situated away from the coding area called as? We need to know what is meant by a promoter sequence, what is meant by an enhancer sequence. Every gene has a promoter, just the way every great man has some source of inspiration. So the promoter is that area of the genome where it acts like a binding site for all the basic transcriptional proteins like the RNA polymerase, etc., etc. Every gene has got a promoter. Now a little away from the promoter site we have another area which is called the enhancer area, and typically this enhancer area contains short stretches of DNA. And why do we require enhancer area? It is the area where specific transcription factors which are making the process of transcription to go on and on, they come and bind with the enhancer area. RNA polymerase binds to the promoter area is what we need to remember. Now the starting point of the transcription corresponds to the 5' end of the mRNA nucleotide is what need to be remembered. And from that point onwards we basically calculate the position of the remaining part of the DNA as +1, +2, etc., etc. And the nucleotide of the promoter, which is adjacent to this transcription initiation site, from there we start numbering it as -1, -2, etc., etc. So what are the important properties of enhancers is a very important question. Basically enhancer is located away from the promoter by about certain length on the genome. Number 2, it can be upstream or downstream from that of the promoter. It can be oriented on either 3' end or towards the 5' end, and it is a heterologous protein, and it enables, it is heterologous in its structure. It enables the binding of the various transcription factors, and this is the important things to note about the promoter sequence. Now what is meant by brain fingerprinting? We have hand fingerprinting. Brain fingerprinting means basically it is the electroencephalogram which is connected. Whenever we are lying, EEG will show some waves, that is the philosophy behind it. When we are truthful, then EEG will show different waves. So probably having a DM neurology spouse in the home, she can put you as a test of infidelity, connect you to the EEG machine and then find out which waves are coming today, so that she is very sure that you are still very much with him or her. So brain fingerprinting is by the interception of certain waves. Not guilty, typically this is the pattern. If the individual is guilty, typically it is the pattern. Based on that, it's called brain fingerprinting. Now let us talk about the important properties of the DNA structure, Watson-Crick's model of the dual helix of the DNA molecule. It is the phosphodiesterase linkages which are the ones which are typically the bridging linkages, and the strands are basically aligned in a anti-parallel fashion from one another. One is in 3 to 5 direction, other is in the 5 to 3 direction. That is how they both are aligned with each other. Adenine binds with thymine. There are 2 hydrogen bonds between the two, and the guanine binds with the cytosine. There are 3 hydrogen bonds between the two. Another common question in the exam. And at any given point of time, the Chargaff's rule, the guanine plus adenine is always equal to the total thymine plus cytosine put together is what need to be remembered.